right, it is January the 15th, and it has been, believe it or not, for us, a very short amount of time to recognize it's been a month since we were able to come back to what in fact is an important monthly lab that we do in environmental science related to population. This is specifically something that we'll be connecting with content in Chapter 5, Section 3. But we set up earlier this year, as we should, to allow these populations, and in fact we have two different populations going on here at the same time. Once again, our study species is the mealworm. We have a population that we are in fact keeping the same to serve as our comparison contrast, and we call that the control colonies. We have two buckets of them. They're exactly the same. They have the same number of mealworms, the same type of bedding. We call it the control colonies because they're getting kind of normal food. So what Mr. Brandon and I did is put out some of the oats. These mealworms are in fact as an immature form of a beetle designed to go ahead and feed on dry legumes and grains like oats. So that is a population split between two buckets. That's like we need that for replication. The second population is a treatment population that is, once again, in two buckets. That gives us replication. We have what's called treatment bucket number 1, treatment bucket number 2. Once again, set up at the same time, same source of mealworms, same amount of bedding. We feed them the same amount by volume, but the difference is what we're feeding them. So what we are doing is we're doing a confirmatory study in this class in which we are following up on some recent research to suggest that mealworms will in fact somehow extract energy from styrofoam that allows them to carry on and complete their life cycle. What we want to know is that really true? And if it is, are there any differences between these populations? What Mr. Brandon and I are going to do here in a minute is we're going to randomly sample from all 4 buckets. So the 2 buckets for the treatment side, the 2 buckets for the control side. We're going to go ahead like we have done. We will have a count for the total number of individual mealworms regardless of their life stages, and then what we'll do is we'll mass them as well. But once again, remember that you're going to need to average data from the 2 buckets so that you have the best data. There you go, guys. We will be back with a second video with the results for January the 15th.